This is Radio EBM.
It's like a maze. Like you have to navigate between different turns and dead
Right,
end.
right. When you
Yeah.
solve technically, when you're developing methods, it's one small maze, and you get out, and there's another bigger maze that you didn't
Right.
expect.
Do you have like superstitious rituals at the lab, something that helps you to go straight forward?
No, nothing.
Everyone put a hat on, I don't know, just burn something. No?
Nothing. Yeah, because it's just continuous maze, so you just try in different ways.
Okay. I can recommend using compass and map. That could help.
Yeah, I hope it's working.
I'm checking the chat right now, but I don't think we have like one note over there, but no actual questions yet. Dustin, do you want to read through the Russell's note?
Yeah, I can go read through it. Russell Grant says, this brings up the general gap between certified reference materials and actually being able to either send minimally processed samples to the lab assay for value assignment versus having something listed as meeting the technical standards to be a reference method procedure, but it's inaccessible. Or having minimally processed samples sent from that facility to your lab to be able to have an assignment. So a lot of times when they have a reference method, like general population is not being able to have access to it. And there is a, matrix effect issue that you have to absolve. And we, as they say, minimally processed samples would be something that would be able to use on your instrument and double check your calibration against. So I guess the question is, how do we solve that gap? Does anyone have any, and do you have an idea of how you might want to think about that gap? How do we get samples into the clinical labs from a reference lab or some type of material that can be certified and then sent from these reference methods and be accessible?
I think it's very important topic, like a GCT lab, because most of the method who developed, they like a voluntarily develop, but most of the labs can run it for a long time. Even they register the method as a reference, and you going back, you try to find who can collaborate with you. Sometimes they're not running. It's a problem of funding also, I think. It's like because mostly what we have like a funding fancy things, but it's kind of long term. It's sometimes can be not well funded. It's one issue. It can be, and long term. It's kind of long term stability of the funding as well, also important. And another thing here, what I can say about process material, this is what we try to also convince the people who working with that most of the time it's also important. The issue for lack of standardized assays. Not commutable material. What is a commutable material? It's not like a behave like a patient samples. Most of the time, it's ampoule. You open ampoule, it's certified reference material or something, right? Calibrator, and then you dissolve in some PBS solution, for example, in buffer, artificial. So eventually, it's not real blood, it's not real serum. But then, if you run about different clinical assay, you can see the discrepancy between, because different methods can have different kind of behavior, on molecular level. So and what we try to do, we're pushing that with our C-peptide method, we try to provide the properly calibrated, not only properly calibrated, but also properly commutable matrix. It's a serum matrix. And we try to work with like a companies or the other labs, and we try to provide these calibrators. We say, oh, you can use our calibrators. They work because it's a patient sample, it's like a serum. It behaves the same way. So eventually, in what we do, like some other published study, so if you use a proper commutable matrix, eventually every assay become like a much closer to each other. If commercial assays start to use them, eventually we can get uniform results, and then it can be some of the problem can be solved.
Yeah, right. So you basically see that your method will be used not only for C-peptide, but also as a model for standardizing other peptide biomarkers. Is that what you're saying?
Not method, our kind of approach, but it's not we developed this approach. It's kind of already accepted, but because it's so multi-stages, multi-step, sometimes it's difficult to implement practically. The main goal here, that if you do calibrators, it should be proper calibrators. You cannot use artificial because sometimes they're working, sometimes not working. You know, it's like a very also kind of